I'm very pleased to introduce the third speaker. That is Dr. Jaroslav Rokitsky from the Center for Research and Education in Forensic Psychiatry in Oslo in Norway. Give him a warm applause.
Thank you. So first thing, I'm doing this presentation on behalf of Dr. Megan Campbell, who couldn't make it today. So I initially apologize for the decreased quality of this presentation. What I will do, will try to present it as best to my abilities. And no, I do not have any conflicts of interest. So let's start with introduction. And we know that antisocial behavior is a complex phenomenon influenced by multiple factors, including male sex, age, previous crime history, but also substance use and psychotic disorders. And when we put our individuals to scanner, and then we find some aberrations in brain. So those come from two sources. One is genetics, and one is environment. And it's quite difficult to disentangle, because they are already adult, what really caused these things we are observing. So in our study, we decided to take one step back and then one step further and look only at genetics. This way, we tried to, control for the environment as best as we can. So why I dare at all to look at the genetics? So first, antisocial behavior is heritable, and heritability of antisocial behavior varies a bit by whom is measuring it, but in general, its aggressive behavior, aggressive subtype is a bit more heritable as compared to rule-breaking subtype. It's based on twin and family studies, and overall heritability of antisocial behavior is approximately 50%. Then if we would look purely on the genetic studies, so common variant-based heritability, or also known as SNP-based heritability, is 7.7. It's usually lower than the family-based. It's a known phenomena of the missing heritability. Yeah, so we come to the year 2022, and Jorim Tilbek, he publishes a GWAS, a genome-wide association study. And he's not first to publish GWAS, but he's first to report a significant locus in this GWAS. This is how it looks. And it is actually associated with a gene FOXP2. And this gene in particular is related to language development. Further, what he does next, he also looks at some genetic correlation, and he shows that antisocial behavior is correlated with depression. It is correlated with ADHD, also negatively correlated with educational attainment and cognition. But I think we all realize that this is not a monogenic disorder. It's not one gene which needs to be fixed, I based this claim on the fact that all complex disorders are polygenic. It means that there's a lot of effects which are spread all over the GWAS, which make the influence, very small individual influence on the final phenotype. So the first question we ask here, what we are looking here for, like when we will be happy. So now we have one locus, and how many we want to see more? We want to see more, 10 more, and then we will say, okay, we are satisfied with this GWAS. Do we want to see 100 more or 1 million more? So this will be the first part, which we will try to answer. So hence, I will now introduce our approach. So first part, we will try to use a mixer, which I will introduce in a second, which allows us to estimate polygenicity, discoverability. Then we will look at some genetic correlations with the other phenotypes which were not covered in the original paper, and we will try to interpret our results. So mixer, so first I will say mixer is developed by our colleagues in the precision psychiatry group at the University of Oslo, and its code is freely downloadable at this location at GitHub. And in a nutshell, what it does, it divides, so like traditional way to assume jivas is that everything is important. Everything is important. I think it's called infinity small model. So some have very small effects, some have larger effects, but all the SNPs are important. SNP mixer just, relaxes this by saying that no, there is a part of the SNPs which are important and the part which is not important, right? So also it allows us then to estimate this polygenicity, so how many of the SNPs will be causally related to phenotype, how easy to discover them, and finally it can say us how many individuals do we need to gather to discover all the SNPs we want. So let's go to the results. And the answer is 6,000. I guess that this number doesn't make much sense of you because you need some baseline, right, for being able to interpret it. And this 6,000 actually, if we compare to other mental disorders, it's a bit less polygenic. Nevertheless, it's more polygenic than different brain phenotypes like total cortical area or mean cortical thickness. It's more or less similar to ADHD, which it's also strongly correlated, but I also here want to know that it's also very correlated to depression, and depression is much, much more polygenic. Now, how it's easy to discover, how fast we will discover those SNPs. So to discover those SNPs, here is adaptation of the figure from the beautiful paper of Van der Meer, where he also describes mixer, and it would be more or less here. So what does it mean here? So on x-axis we have polygenicity, or how many SNPs are causally involved, and on the y-axis, the higher you go, the easier it is to discover those. So antisocial behavior is approximately more or less the same as ADHD. So to translate it to human language, if we look this on the, if we will look how many people do we need, so now we are here. So this is on x-axis we have sample size, and on y-axis we have proportion of the variance explained by the significant SNPs out of the total variance of the jivas. And we are now at the proud 0.0%, and we will, if we want to explain 90% of the variance, then we will need to have at least somewhat 50 million of individuals. Now we have 8,085,000. So it seems it will take some time for us, but we are a bit impatient. Therefore, I just use the jivas we have right now for my future analysis, for my further analysis. So first thing what we did is we looked at the genetic correlation with other disorders, and here we see that it's genetically correlated with depression, which is good, which was also in the original paper, so it would be surprising to find something else. But also it's correlated with bipolar disorder, but not correlated with schizophrenia, which is a bit surprising, taking there is a number of epidemiological studies across Scandinavia which show that schizophrenia, the patients with schizophrenia are at increased risk of perpetuating violence. Further, it overlaps with most of the substance use, both frequency of use and also disordered use. But when we go to the brain area, finally, suddenly we do not have any significant results. It would be very wrong to me to say that those two things are not related, because I can't say this. But if any of you have ever done the studies on antisocial behavior, you probably know what small effect sizes we have and how easy those disappear once we start to control for different environmental variables, right? So maybe this is a bit of explanation why this happens. Yeah. What would we do next? Okay, let's. Oh yeah, and this is those structures which, if you will read literature, those structures will reappear in the. You will find them in other papers. So next thing which we did, we mapped this genetic correlation on the brain. So thickness, so it's a genetic relation between these brain structures and cortical thickness, and cortical area and cortical volumes. So here what I found interesting is that cortical thickness is associated with so antisocial behavior is associated with decreased prefrontal cortical thickness. Logical, yes. But when we look at the cortical area, all of a sudden it's associated with increased cortical area, which I don't know like how to interpret. We usually like to interpret negative things with decreases. So when I tried to look for some interpretations for this, I stumbled upon this study which was conducted in China, and here they scanned people with antisocial behavior who were recruited from school jail. So they were scanned when they were already adults, but they spent some time in school in a jail. And also they find a very similar pattern. So they report decreased cortical thickness and increased cortical area in that sample. And even I would say that the patterns quite match our findings, but this is a very small sample. And as you know, it's very easy to cherry pick a study which would confirm your findings. So just contradict myself, because I like to contradict myself, is this study by Enigma Antisocial Group, where they looked into conduct disorder, which is a precursor for antisocial behavior. And here they show that actually conduct disorder is associated with massive decrease in cortical area. And I would recommend you to trust this study more, because it's a lot of Enigma people here around walking. And the last thing what we did is we took neuromaps. So neuromaps is a like resource where they like gather the different maps across different studies. They have in total 73 maps, and those represent multiple things. They represent electrophysiology, they represent development, they can represent gene expression, for example, so like, but like very globally, like very crudely. For example, they can represent the first principal component across all the gene expression from Allen Brain Atlas map. And then we correlated them, because we just, the basis of the study is running correlations, never enough. And what we found is that this brain map of cortical area was positively correlated across the 73 maps, most positively correlated with glucose metabolism. And fun fact, if you would put this GWAS, original GWAS, you would annotate it, you would really find a lot of hits on different metabolic measures, BMI and so on. But in a nutshell, what it's negatively correlated is serotonin dopamine transporters. And the same with cortical thickness. When we tried to look at this, we found that it's negatively correlated with serotonin receptors. And so like all this theory between the antisocial serotonin deficiency hypothesis, which suggests that reduced serotonin function predisposes you to aggressive and antisocial behaviors, can be a bit maybe far-fetched based on these findings, but at least, I mean, if you remember when I was showing the genetic correlations with different disorders, it was correlated with other mood disorders like depression and bipolar. So, I mean, at least what the study shows us, because these findings are, most of these correlations are not significant, right? But at least we circulate constantly around this same topic, right? So it's another evidence that this antisocial behavior is related to depression and, in general, mood disorders. So, some take-home messages. antisocial behavior is moderately polygenic, but larger N is needed to discover majority of associated variants and overlap. So we found overlap between antisocial behavior and mood disorders and substance use, and it has lower cortical, it's associated with lower cortical thickness in prefrontal and temporal cortex, and shows inverse correlation with serotonin and dopamine neurotransmitter maps. With this, I want to thank you all the co-authors, and especially Megan Campbell, who essentially presented this for you. Thank you.